In Lecture 1, we covered topics as to why we are interested in conducting clinical trials, the history of clinical trials, and some of the ethical and federal regulations governing the conduct of clinical trials. We're going to talk specifically now about what's a clinical trial and why we should bother conducting clinical trials. To define a clinical trial, it's an experiment in humans for the purpose of evaluating one or more potentially beneficial therapies where the investigator has control of some aspects of the trial as he's designed it. In this slide, I've indicated sort of a general flow of the inference we want to make from the studies we conduct. On the left-hand side, I have a box labeled patient population. This is the population we ideally have in mind about evaluating whether the therapy would be useful for that population. We will go through some of the basic elements of defining a protocol, a research plan. And part of that plan, as we'll get to in later lectures, defines the entry criteria for patients that come into the trial. Not every patient that we have in mind will be suitable or ideal for the trial we're going to conduct for various reasons. They may have contraindications, they may not want to, may not be in their best interest, and so forth. So we define entry criteria. However, we will get a sample of patients that actually show up, the volunteers, those who consent, and they will not necessarily reflect completely the entry criteria we specified in our protocol. They will be the ones who showed up, not necessarily the ones that we had hoped for in our study. We observe results on those patients who showed up, our patient sample. And at the end of the trial, our goal is to make inferences about the results we observed on the sample population that we studied. That's what statistics is going to do, is make inferences about people who look like the ones we saw. There's more of them. However, we would like to make inferences, what I call inference B, at least on the population that we had in mind in our protocol. But we may not have studied all of those. So as we move from the right to the left, inference A is pretty solid. That's the standard statistical inference. Inference B is a little less solid because not all the people that we've defined as eligible in our study, our research plan, actually showed up. And finally, we'd like to ideally make inferences to the patient population we have into our clinics or into our hospital. We didn't study all of those. So you have to be careful as you make inferences from the research you conduct. You can certainly make inferences about the kind of people that you actually studied. But as you go to the next level, you're making a bigger stretch, and finally to the population making a huge stretch, typically. We talk about different kinds of trials, single-blind trials, double-blind trials. By a single-blind trial, we mean a trial where the participant, the patient, does not know the identity of the treatment being received, but the investigator does. In a double-blind trial, neither the patient nor the treating physician or healthcare team know the identity of treatments being administered. Now, not all trials can be double-blinded or even single-blinded. Obviously, a surgical trial would be more challenging to blind than a drug trial. If you're an ophthalmologist, of course, you would not use the term single-blind or double-blind, but you would refer to it as a single mask or double mask. By a placebo-controlled trial, we need to be careful about what we mean. A placebo, of course, is an inert substance made up to physically resemble the treatment being investigated. It's usually used as a control treatment. Now, the design, as we'll talk about in a later lecture, may be comparing a treatment versus a placebo, as the patient is either getting a treatment or a placebo. But more than likely, it's the best standard of care for the patient, plus either the experimental treatment or a matching placebo. There's often been comments that patients aren't being treated at all, they're given a placebo. That's probably not the case. They're probably getting the best care we know, and either a new therapy or a matching placebo. We talk about different phases of trials. I alluded to this earlier in the lecture 1. The phase 1 trial in humans is usually the first in humans, and the aim is to find an acceptable range of doses and schedules of the drug or the biologic. Even for a device trial, there is the first in humans experience where you understand some properties of the device. Phase 2, you now want to move on and say, if I give this therapy, this drug or biologic, at this dose, do I get the results, the intermediate results that I want? That is, does the drug reduce blood pressure or cholesterol? does the device behave in the way we intended? The third phase we call the comparative trial. Here you're trying to establish, at this particular dose or use, does the therapy deliver the clinical benefit that we are intending or hoping for? It is sometimes referred to as the definitive studies. We also refer to phase 4 trials, and this is being given much more attention these days in post-approval trials, where we attempt to assess in longer-term surveillance safety of the device or the intervention. Phase 4 trials are challenging because they're typically not randomized. They are observational in some sense. And so much work needs to go into the phase 4 trial because the current standards are not all that adequate for the needs. We talk about randomized trials, non-randomized trials, single-center trials, multi-center trials, and different phases. So there's lots of language that you have to pay attention to as you read the literature as to exactly what kind of a trial are we looking at. Well, why are clinical trials needed? We believe, and think we can argue, that this is the most definitive method of determining whether the treatment is effective or not. As I've indicated, there are many other designs where we learn things about a new therapy. But these other designs have more potential for bias. Bias means that one therapy has inherently an advantage over the other by the nature of the design. Bias is very powerful, and a hard lesson to learn is that large numbers, large studies, do not necessarily reduce bias. It reduces noise, but it may not reduce bias. And the frustration is that one cannot determine on the basis of an uncontrolled study whether the intervention has made a difference or not because of biases and confounding factors. There are many observational studies that have suggested therapeutic benefit. But the issue is that correlation is not the same as causation. There are many examples of false positives, that is, associations that were suggested to be the cause. But in fact, with later research, turned out not to be the case. You can probably add many examples of your own to this list. We also believe in replication as being important in science. And clearly in the field of lung cancer and smoking, there were many studies which were able to replicate high risk of lung cancer among smokers. But replication by itself is also not sufficient if fundamentally there's confounding and bias. A very interesting example is the studies of beta-carotene and their association with cancer. Many observational studies showed that low levels of beta-carotene were associated with high incidence of lung cancer. Yet when trials were conducted, increasing the levels of serum beta-carotene, they in fact did not reduce the cancer incidence, but in fact increased it. So just because studies are replicated doesn't mean we still have gotten the cause and effect answer. Another reason for clinical trials, we like to determine the incidence of side effects and complications. An example from the Coronary Drug Project, one of the earliest cardiovascular trials conducted in a population of men who'd had a heart attack. Drugs were given. 2 of them are listed here, clofazimine and niacin, to lower levels of cholesterol and hopefully reduce the incidence of cardiovascular disease, death and MI to be more specific. Nevertheless, when you examine these drugs for their side effects, you'll notice in the area of cardiac arrhythmias that clofazimine and niacin had a 33% increase, or incidence rather, of cardiac arrhythmias. One might think that that's pretty alarming. However, in this population, the placebo arm had a 38% incidence of cardiac arrhythmias. That is, this problem, this side effect, is a function of the disease and the disease process in that patient. Without the control arm, one would never have been able to ascertain that the cardiac arrhythmia incidence in these 2 drug arms was just consistent with the placebo arm. Similar for nausea. The clofazimine arm reported an incidence of 7.6% nausea. But the placebo arm reported an incidence of 6.2. So we need controlled studies to determine whether the side effects we see are related to the therapy or just part of the disease process. There are many studies, therapies, which have been accepted with no trial. Later, we find out through a trial, perhaps, or through some other means, that the therapy, in fact, was not useful or potentially harmful. Some examples are listed here. The IPPB trial, or the intermittent positive pressure breathing trial, was a trial in patients with chronic obstructive lung disease. A device, which under pressure delivered bronchodilator therapy to these patients, was used to reduce exacerbations from their problem. This therapy became in widespread use. It was expensive, yet there was no trials that demonstrated benefit. Finally, the IPPP trial was conducted versus best medical care and indicated there was no benefit in many dimensions that were examined of this device. Similarly, for the use of high dose of oxygen, an example which I'll take a more careful look at in just a minute. Many other therapies become in widespread use without trials, and we discover later that they were not useful or potentially harmful. A new area that we can think about why trials are needed is to evaluate new frontiers. Today we think about genomics, or perhaps proteomics. But this offers a whole new dimension to examine diagnostic trials using genomic profiles for diagnosis of disease, for prevention of disease, for gene therapy trials, gene transfer trials, and maybe, hopefully, to use genomics to speed up the drug development process. So we have a new frontier here of genomics and proteomics that the clinical trial will be an exceedingly useful tool to sort out what aspects are useful and which are not. Now, sometimes we practice or follow a practice based on theory. Let me share you a little more detail the retrolental fibroplasia story, a nice publication by Silverman in 1977 in the Scientific American. High-dose oxygen for premature infants became the standard of care. The lung is the last to develop, and if a premature infant has problems with their lung capacity, high-dose oxygen should help it along. That was based on the theory. However, it was observed at the same time that there was an increased incidence of blindness. Pediatricians became interested in this and searched for the cause. While the high-dose oxygen was suspected based on some anecdotal evidence, there was other studies, observational studies, that said no, it was a benefit. When one has such a controversy or an uncertainty, it suggests the need for a controlled clinical study. To show you how strongly theory can influence practice and people's views, one study was attempted to examine the effect of lower dose oxygen. But in the first attempts, the nurses felt that not giving high dose oxygen was unethical and not in the best interest of the patient, went in at night and turned high dose oxygen on and then off in the morning. So it defeated the purpose of the experiment. The problem continued, and in 1953 the NIH held a conference. Two opinions emerged. No controversy was settled. One said we need to have a study to sort this out. The other opinion was no need. High-dose oxygen is already convicted as the cause. Well, a trial was conducted on 800 infants, the standard dose versus half of the standard dose. And looking at the incidence of blindness, it was 23% for those given the high dose, the standard high dose, versus only 7% for given the lower dose. These results were published in 1954. The high-dose oxygen practice was stopped, and the epidemic subsided. However, meanwhile, it was estimated over 10,000 infants had probably been blinded by this practice. Sometimes the opposite happens. Sometimes we fail to use a therapy based on theory. Chronic heart failure is a major problem in heart disease these days, and in the 1980s, there were not many good therapies for treating chronic heart failure. There was a class of drugs called beta blockers that were known to be effective in post-MI myocardial infarction patients. It's a drug that reduces mortality, lowers blood pressure, it regulates the heart as it slows it down. And for a heart failure patient, where you already are having problems with the heart being efficient, these kind of properties seem like the wrong thing to do, just the opposite. And so using a beta blocker in a heart failure patient was proscribed. It was not recommended. Some investigators persisted in examining this question and developed enough preliminary data to suggest that some clinical trials of beta blockers in heart failure be conducted. Several trials were launched about the same time. In class 2 to 4 heart failure, this is ranging from patients who have some limited ability to conduct their daily life to severe limitations in conducting their daily life. These patients had low ejection fractions, that it was a beta blocker versus placebo trial. They were randomized, double-blind studies involving several thousand patients with mortality as the outcome. That is, would a beta blocker reduce mortality in this population? I want to share with you 3 studies that were all conducted within roughly the same period of time. The first is a trial called MERIT, a trial of several thousand patients, 4,000 patients roughly. And what I've shown you here is the mortality curves for the 2 therapies. The placebo arm obviously has a much higher mortality rate than those on the metoprolol arm of the study. Metoprolol is one of the beta blockers. In fact, a 34% reduction in mortality, a highly statistically significant result. A second trial called the CEBUS trial, CEBUS 2 as a matter of fact, also using another beta blocker, bisoprolol, similar design, demonstrated a highly significant reduction in mortality for those on bisoprolol. Here is the Kaplan-Meier survival curves, confirming the results of MERIT. A third trial conducted in the class 4 patients of heart failure, the more severe patients, using a third beta blocker called carvedilol, also demonstrated an improved survival with patients on carvedilol compared to those on placebo. Again, highly significant. So here we have 3 trials involving 3 beta blockers versus placebo, all demonstrating significant improvement in survival compared to placebo. Yet, for a decade or more, we believed, based on the theory, that these therapies should be prohibited from being used in the chronic heart failure population. Theory proved us wrong. Sometimes we have data, long-standing therapy, based on both theory and observation, or associations. A classic case of this is the hormone replacement therapy field. A hypothesis was accepted that the use of hormone replacement therapy in postmenopausal women would reduce the incidence of coronary heart disease. There was lots of supportive data. We knew that hormone replacement therapy lowered lipid levels. There was non-human primate studies. There was a lot of observational studies. That is, women using hormone replacement therapy had a lower incidence of heart disease than those who did not. These are some of the references, a few of the references that provide that evidence. These studies suggested a substantial, a 40 to 50% reduction in coronary heart disease risk for women on hormone replacement therapy. As a result of this kind of data, HRT use became quite popular. In the year 2000, it was estimated that 1/3 of postmenopausal women used hormone replacement therapy. It was the second most prescribed drug. 40 million prescriptions for the estrogen and 22 million for estrogen and progestin. Over $1 billion in sales. Yet there was no, at that time, no available randomized clinical trial data. 2 trials were conducted in a similar period of time. One is a secondary prevention trial and one a primary prevention trial. 2 labels, again, which I need to explain. Primary prevention trials are trials where we want to prevent the event from ever occurring. Secondary prevention trials are trials in which we want to prevent an event from happening again. So if you had a heart attack, we want to prevent the second heart attack. So these 2 trials that I referred to here, one is called HERS, the other is called the WHI, or the Women's Health Initiative, were conducted. The HERS trial, published in 1998, was in postmenopausal women who already had some documented cardiovascular disease. It was estrogen, progestin versus placebo, randomized double-blind. Its outcomes were cardiovascular mortality and fracture rates. It was already established that hormone replacement therapy would reduce bone loss in women, postmenopausal women, and therefore reduce the incidence of fractures. In the conduct of HERS, it was observed early that there were clotting problems. Deep vein thrombosis, or DVTs, pulmonary embolism, both had an increased incidence. As a footnote to our discussion in Lecture 1, because the committee that was monitoring this study observed these results, it led to an early disclosure to investigators and patients that there was an increased risk of these clotting problems and that women, for example, who were mobilized should probably be taken off of HRT during that period of time. The trial demonstrated, as expected, a fracture benefit, or at least a trend for a fracture rate benefit. But on the cardiovascular side, There actually was an early negative trend in mortality that is suggesting harm, but as the trial went on, it eventually reversed to neutrality, and the result was in some sense not definitive. Here I've shown you the mortality results in HERS over the period of time of the study, and you can see the early negative trend as a higher incidence of mortality, but converges as the trial continues in its 5th year. The impact of HERS was rather interesting. Many people believe that these results only apply in secondary prevention, as their belief in HRT as a primary prevention remained intact. In fact, many interpreted the trend, the reversal in the trend as the trial went on, suggested that if you only would have gone longer, you would have seen the benefit. So as a result of HERS, there was no perceived impact on the use of HRT, since HRT had other benefits, symptom relief, fracture rates, and so forth. It really didn't change the use of HRT much at all. A second major trial, actually a series of trials in one, was published early in 2002. The Women's Health Initiative, or WHI as it's known, was a large factorial trial. We'll examine what factorial trials are in a later lecture. It was evaluating in the same study the use of HRT, low-fat diet, and calcium supplements. For each treatment, there were several outcomes that were being assessed. For HRT in particular, we were monitoring the incidence of coronary heart disease, both MI and coronary heart disease death, the incidence of invasive breast cancer, a global index which would serve all the good things and bad things that could happen in one index, and finally, fracture rates. It was, to date, the largest trial in women ever conducted. You will notice by this diagram that over 370,000 women were screened as being eligible for this trial. 18,845 patients were, in some sense, eligible by providing consent and had it in this arm of the study had an intact uterus. 16,608 patients were randomized, half to the treatment arm and half to the placebo arm. And you see here on the status of those patients as of April 30th, 2002. I want to show you here a series of what we call Kaplan-Meier graphs for the incidence of these events. As you can see here, over the 7 years of follow-up, it's on the x-axis, and the cumulative incidence on the y-axis, that the global index, sort of the overall gestalt of all the good things and bad things, is, roughly speaking, the same, although a slightly higher increase, those on the HRT arm, but not significant. Mortality on the right-hand panel is nearly identical, with a little bit of splaying at the end, but those represent a small number of events on a smaller number of participants. However, if we look at some of the other outcome measures, we notice that for coronary heart disease on the left panel, that the HRT arm actually has a higher incidence of coronary heart disease, in fact, a 29% increase. HR, the hazard ratio of 1.29, indicate that an increased incidence of coronary heart disease. Likewise, the other concern was the impact of HRT on invasive breast cancer. And as feared, over time, the incidence of invasive breast cancer increased. In fact, there was a 26% increase in the incidence of invasive breast cancer for women on HRT compared to placebo. Pulmonary embolism, one of the indices of clotting problems, was also increased for women on HRT. In fact, the hazard ratio, or the incidence, it was 2.13, over a doubling in the incidence. Stroke, another indication of clotting, was increased by 41%. Colorectal cancer, on the other hand, to some surprise, was reduced. In fact, the incidence is 63% of the control rate, a reduction of 37%. Hip fractures, as expected, were reduced as well. So the concept that HRT reduced incidence of fracture rates was confirmed. It was already believed and reasonably well established. So we confirmed that. The incidence of colorectal cancer, the improvement was one that was not expected, I don't believe. And as many trials, you find new issues that you're not sure of, which need to be confirmed in later studies. The lesson here, I think, is that a therapy based on observational data, based on theory, it became widespread practice, among the largest drugs prescribed, unknown to us, had some serious harmful effects on coronary heart disease, on stroke, on DVTs, pulmonary embolisms, and on breast cancer. Again, theory and observation gave us the wrong answer. This, of course, was a shock to the medical world and to the postmenopausal women who were taking the therapies. It was not expected. In fact, some thought perhaps the trial was unnecessary. Many editorials were written. What went wrong? Well, as this editorialist writes, we shouldn't be so surprised. We took a shortcut and accepted the therapy without putting it to the test, as we based our practice on theory, on intermediate results, on observational data, which is subject to bias due to confounding in ways we can never fully explain. But the bottom line is we never tested this therapy in a controlled, rigorous study. So we believe in evidence-based medicine. For the most important questions with serious outcomes, mortality, morbidity outcomes, we really do need to conduct well-controlled, randomized studies. If it's not possible to conduct a trial, and there are many situations that it's not possible, we nevertheless need to be cautious and vigilant about the treatments if they're based on observation or theory. We need to understand the level of evidence on which we're basing our practice. A challenge is, when should we start a trial? There's never an ideal time, but there's some things we need to know before it's reasonable to start. One is we need to have enough understanding of the intervention, the drug, the device, the biologic. Something about its safety. We call the Nuremberg principles. You wouldn't knowingly put someone into a study if there was known safety problems. We need to understand the correct dose and the duration. Knowledge of, do we have the final form of the therapy? The little footnote, the TPA story, is we were studying a therapy, TPA, a clot-busting therapy, in which the preliminary data indicated that this therapy worked really well. We were then set to do a large study, and the manufacturer, the sponsor, sort of scaled up production to produce more of this material. And in the scaling up process, they inadvertently or unknowingly changed the potency of the product. So when we went to the major trial, the definitive trial, with the new product essentially, it had much higher potency, and immediately we began to have bleeding problems that we had never seen in the preliminary pilot studies. So the lesson from that was that we did not actually have the final form of the therapy we're going to use. And in changing the production process, the potency was changed unknowingly to us. We have to define the study population. If we wait too long, the therapy may become obsolete. This is a problem in device trials because the engineering keeps changing. In the field of drugs, the new drugs keep coming out with different characteristics. In the trial design, as we'll talk about in the next lecture, we need to understand exactly how to measure what's important. Can we measure it? Is it the expected effect of the intervention? These are really serious questions. Thirdly, feasible. Do we have the financial resources, the staff resources? Is the equipment and technology available? And are the subjects available, most importantly? One of the ethical issues is one should not start a trial that you can't potentially finish. Why ask patients to participate in a trial that will never be completed? There's a serious ethical issue inherent in that. There is a very narrow window in time in which a trial can be done. If you do it too soon, things aren't stable. The first and earliest trial of coronary bypass surgery, the so-called VA bypass trial, there were some early operational deaths. And the criticism was, well, the surgeon was still learning and mastering the technique of bypass surgery. So it was not yet a stable procedure. On the other hand, if you wait too long, the window closes and the intervention becomes accepted practice, whether proven or not, but it nevertheless prohibits or certainly inhibits your ability to test. The bypass surgery trial, for example, we talked about the VA trial being done too early. A few years later, the NIH started a trial called the Coronary Artery Surgery Study. It was a randomized trial of surgery versus best medical care. However, the window was almost closed because less than 1,000 patients were randomized in that trial, but several thousand patients were put into a registry because people believed at that point that bypass surgery worked. The PTCA, or percutaneous transluminal angioplasty, was a window that opened and closed very quickly, and no trial was ever done that compared PTCA versus best medical care. What was done, for example, is comparing PTCA versus bypass surgery. So we need to know enough to be able to design a good trial. If we wait too long, the therapy becomes practice, whether proven or not, and then it's very difficult to conduct the trial. A key principle which I want to introduce now, but I'll come back to in a later lecture on the issues and analysis, is something which I will call, and is called, the intent to treat principle. It's an incredibly important principle. Simply said, it says that you must analyze or account for all subjects that are entered into your trial, regardless of the compliance to protocol. This principle has a terrific impact on how we design trials, how we do the sample size, how the trial is conducted and analyzed. It's the primary analysis for trials, and as you learn in later lectures, one thing that you need to look for is you read and review and critique a trial. There are many other analyses that can be proposed, something that's called per protocol, which means you analyze the patients who complied to the protocol or analyze them while they were taking the prescribed therapy. That it must be always a secondary analysis. One example or issue that comes up is that sometimes investigators believe that if the patient stops taking the therapy, stops taking the drug, you can stop following the patient. That practice is in violation of the intention to treat principle and really introduces serious potential bias. We'll come back to that in a later lecture. But I want to introduce the idea that as we go through the lectures, I'm always thinking about this ITT principle that I must account for all subjects that I randomize and enter and all events that happen to them until the trial is closed. So I'd like to close by discussing a little bit the organization of a trial, the structure of it. What I'll share with you is the structure of some of the larger multi-center trials that you read about typically in the literature. But even if you're conducting your own study in your own clinic, you will still need the components of this structure in order to successfully complete your study. One of the first trials that were conducted was by the National Heart, Lung, and Blood Institute. They're preparing themselves to evaluate the risk factors that they had identified through the Framingham Heart Study, i.e. blood pressure, lipid levels. So they convened a group chaired by Dr. Bernie Greenberg, and this report of this task force became known as the Greenberg Report. It was published finally some 20 years later. But it identified some of the key components that you needed to conduct a trial. Obviously, you need a funding agency to pay the bills. But more importantly, they identified that you need some steering committee, some leadership to write the protocol, to monitor the conduct of the trial, to analyze the results, write the papers, do all the work that needs to be done in conducting a trial. Of course, no trial can be conducted without a lot of help. So in the bottom row, I've listed some of the things that they identified and suggested. There are many working committees that come up in designing a protocol, the eligibility criteria, the compliance committees, the assessment committees, and so forth, that need to be put together to come up with the study design. Lots of laboratories will be used to measure blood lipids, electrocardiograms, EKGs, for example, or X-ray assessments. Patients, of course, and the clinics are the heart and soul of a clinical trial. Data from that clinic needs to be coordinated in somewhere, so we've identified a data center or a coordinating center. We'll come back to the big box on the left a little bit later, but the Greenberg Report, as do the Nuremberg Principles, the Helsinki Declaration, and the federal regulations now, said you must be prepared to stop a trial early if there's evidence of harm. And the Greenberg Report suggested a monitoring process. They actually allude to two components: overall policy advisory board to the institute, to the funding agency, and to the steering committee, and a data monitoring committee. Later in time, these two were merged into a single data monitoring committee. But this is the model that we'll talk about later in the monitoring lecture. But this model gives us a framework for how we should think about how a trial should be conducted and the necessary components. So while this is structured around the multi-center trial, I would argue that in your own center, you need to have someone who is supporting you, whether it's your department or an outside agency, somebody, perhaps just yourself, but more likely a small group, that's responsible for the trial, the protocol, its conduct, a series of supporting groups, and the clinics where the patients are coming from, including a clinic coordinator that is really taking care on a day-to-day basis of the recruitment of subjects. I've detailed here a little more what each of these components are. I won't go through them in detail, but the funding agency obviously has responsibility for the trial. They are the ones that are justifying the funding that's going into it. For industrial studies, they obviously have interest in taking the results of the study for approval with regulatory agencies around the world. The advisory board and the monitoring board is an important component. It's the outside review, the checks and balances in some sense, and was one of the strong recommendations of the Greenberg report. The steering committee or the executive committee is a group of people responsible for the conduct of the study, and they're usually the ones who write the protocol, review the interim results for progress, not for benefit and harm, but for progress, and at the end of the day, we'll get together and write the paper and present it in public arenas. Sometimes we call the assembly of investigators, or maybe the steering committee, depending on how large the study is, but all the investigators need to have some input and communication during the trial as well. The coordinating center is where all the data usually go. It's collected, edited, entered, analyzed, and presented to the various groups that have interest. The laboratories that are engaged depend on the trial. Maybe biochemistry, pathology, EKG, X-ray, or other more sophisticated laboratories. As I said, the grassroots of any clinical trial is the clinic, where the patients come through or participants are encountered. Throughout the rest of the lectures, we'll be examining essentially the components that are necessary to put together a clinical trial protocol or the research plan. It's required for all trials. You will not get Institution Review Board approval if you do not have a protocol. It's the basis of the review for the ethics. It's typically the basis of the review for the funding. So it's a very important document. It is sort of the blueprint for what you're going to do. Some of the background and rationale for the trial need to be included. You need to specify the objectives of this experiment, the primary question, secondary questions of interest, whatever logical subgroup questions can be posed, and finally to understand whatever toxicities and side effects are the result of the therapy. There's a major section of your protocol which defines the design. That includes the population you intend to study, inclusion criteria, exclusion criteria, some rationale for the sample size, a plan for how you intend to enroll patients. One of the critical elements of any trial is the recruitment strategy. It's easy to underestimate the challenges in recruiting patients. You need to define the randomization plan, and we'll examine that in some detail. Of course, you need to define the intervention you're going to study, the frequency of follow-up, and what data is collected at each of those follow-up visits. Especially since the Shalala executive orders, we need to have a monitoring plan for every trial. That plan may be simple or it may be complex, but there's lots of things to monitor. You want to monitor the quality of the data, the progress of recruitment, the risks and benefits, and the potential for early termination if the data are poor quality, the trial is just not recruiting, or the risks significantly outweigh the benefits, or occasionally the benefits overwhelm the risk. You need to identify a plan, the analysis plan, and how you plan to report it. We will talk a lot about you must specify the questions in the experiment before you start. You also need to specify how you plan to analyze the data before you start. Because if you allow sufficient analysis, one might, by pure chance, find something that looks interesting, but in fact it's just a result of data dredging and not some rational plan. And finally, it's a good idea to have some description of the organization, who the investigators are, who the committees are, who's in charge. Another document which sometimes accompanies a protocol is a manual of operations. I sometimes refer to the protocol as the blueprint, but the manual of operations as the construction document. This really drills down to detail at every level about how things are going to be measured, how things are defined, what equipment is going to be used, how it will be used, the data forms that will be used to collect the data, the instructions for completion, and so forth. It should include whatever detail is important to conduct the trial properly. So we've covered in this lecture some of the reasons why we need to do trials, that if we take shortcuts, we can sometimes get wrong answers. Sometimes those wrong answers can cause serious harm, or at their very least, waste resources. We can't do trials for every question, not even every legitimate question. We must choose to do trials of the kind we're going to talk about when the question is really important and the implications, the outcomes are really serious. Next time, we'll be talking about design of trials, and we will look into that in more detail.